My brain says I should drink chocolate milk, but my stomach says, are you sure about that? All right, I'm Miss Norris again, and this is Norris Teaches Science. And today we are going to talk in this biology edition of my video series about enzymes. And your goal by the end of this video, I can explain the process enzymes perform in living systems via a cell or a human body. So first, let's start off with what are enzymes? All right, let me get my laser pointer. Here we go. Enzymes, one, are a type of protein. I have already done a video that talked about proteins. I'll link it in the description below if you need to go back to it or you've never seen it. So it's a type of protein. It's responsible for speeding up the rate of a reaction. That just basically means it makes reactions happen faster. They are very specific. They're very specific about a lot of things. If it's not fitting the specificness, it's not going to work. They're found in many different parts of the body, and they usually end in ASE. If if it ends in OSE, you're looking at stuff that's a sugar, which would be a carbohydrate, and then the ASE is the protein or the enzyme that would break it down. So, example, lactase. Lactase is in your body to help you digest lactose, like found in chocolate milk. Some people who are lactose intolerant lack the enzyme of lactase so they can digest it very well. You also have many other ones like oxidase, amylase, protease. They're all there for different reasons in your body. If you want to know more about all these different reasons, I would check the Google. Now, the enzyme structure, I have a picture over here of an enzyme structure. Is generic. You have the enzyme, and another thing you have is something called the active site. This is this area right that I'm highlighting with the laser pointer. It's the active site. It's where the enzyme binds with the other part called the substrate. That's the pink part up here. And substrates are specific reactants that an enzyme act on. It means they have to be coherent. They have to work together. It's kind of like a lock and key idea. If you have a key to your house or apartment or whatever you live in, and you try to use your key on your friend's door, it's not going to work. Keys are specific to each lock, just like substrates are specific to each enzyme. Now, enzymes' purpose are to lower activation energy required for a reaction to take place. And that's a lot of scientific terms. I get it. It means to make a reaction happen faster by using less energy. And the less energy you use in reactions, the more energy you have for other stuff it wants to do. Say if it's a cell, it has more energy to move than to, digest its food. And so that means that enzymes are something called a catalyst. They're not used up in the reaction, they're just there to help it happen faster. They're like, say you're doing your homework and you're supposed to be coloring something, it's a project, and you have all the information up there, and then you get your little sister or brother to come in and color the pictures you draw. They were the catalyst. They're not, they're just there to help it go along. So, how enzymes work? It's a lot going on in this slide, I understand, but here we go. First off, the reactants or the ingredients are called the substrates. And like I've already said, the substrate will bind at the active site of the enzyme. Then two things can happen. It all depends on what substrate or substrates bind to the enzyme. So if we look at this picture on the right, I'm highlighting the top one shows that you have a substrate bind, and then the enzyme breaks it apart to form multiple products. So it could break apart the substrate. The other one is it could take multiple substrates in. You see this at the bottom. They react and it produces one product, which means it bonds or binds the substrates together to release a product. The final thing is called a product, and this is released. Now, the enzyme is not changed during the process. That's why it's called a catalyst. So once it's done with this process, it can go find more substrates and continue the process along. That would give it time, give it the reason why it's called a catalyst. It's not changed, but it can keep working. But enzymes are specific. Remember, I said they're specific. I already said that they're very specific to their substrate. Only certain substrates work with certain enzymes. But enzymes are very specific to their environment. What I mean is they can only work at certain temperatures and at certain pHs. Each enzyme is different about what temperature and pH it is, but it is that. Side note, if you don't know what pH is, it's how acidic or basic something is. So we have here in this picture, here's an enzyme, here's a substrate. They fit together perfectly in their current condition. Now, if you heat it above, this enzyme is heated above 40 degrees Celsius. What happens is its active site and its shape gets changed. And when that happens, we call that denatured. And so the substrate can no longer bind at the active site of this enzyme because it doesn't fit perfectly. And so that means the enzyme will stop working. So if it's not the right substrate, it won't work. If it's not the right temperature, an enzyme won't work. If it's not the right pH, the enzyme won't work. And temperature and pHs can make the enzyme become denatured. So reviewing the enzyme process, I have a picture over here. Things are labeled, and here they are. A is right here that I'm highlighting with the, what is it called? Laser pointer? Yeah. This is called the substrate, and B is the enzyme. When they combine together and they're locked in together and the reaction is taking place, we call that C, which is the substrate enzyme complex. That means the reaction is taking place. Once the reaction's done, D is still the enzyme. Remember, the enzyme doesn't change. It's a catalyst. And E are the product or products. This case, we were breaking down a substrate into products. It could have been multiple substrates combining. Either way, E is the products. So this is the enzyme information, the process, how enzymes work. There's a lot more out there about enzymes. I just covered some basic information. If you really want to dive further, I suggest you check over at the Google or go read a book. But this is Mr. Norris. I really hope that you learned something, and I am signing out.